எந்தாயிடுச்சா
இதை இழுத்தா இது
நானும் மாதிரிதான் சும்மா போட்டு தேய்ச்சிட்டு அப்பதான் பார்த்தா தெரிஞ்சது அது போயிட்டு தெரியும்.
Hello, hello, check.
Hello.
Good afternoon, ma'am.
Is it audible now, sir?
இதுல சவுண்ட் ஃபுல்லா வச்சுங்க தயவு.
Hello.
Hello, can you hear me? Hello,
Hello.
hello, Dr. Rajendran, can you hear me?
Yes, 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 doctor. Your voice is audible.
Yeah, thank you.
Okay.
Rajendran sir. Rajendran sir.
Hello, sir.
Okay, sir. May I audible?
Yes, sir, you are audible, sir.
Thank you, ma'am.
Thank you, sir.
Hello, Suresh sir.
Hello.
Hello.
Yes.
Yes, sir.
Yes, sir. Shall we start the program, sir?
Yeah, yes.
Yes, we can proceed, please.
Okay. Sir. Hello,
Just
Ashok
arrived,
sir.
right?
Yeah, yes, sir.
Are you ready? Shall we start the program, sir?
Yeah, we can proceed.
Oh. Good afternoon, all. I am Dr. G. Rajendran, Assistant Professor in Department of Biotechnology. I want to start the session with a formal welcome address. So I would like to invite Dr. S. Suresh, Assistant Professor, Senior Grade, for delivering the welcome address. Sir, over to you.
Yeah, thank you, Dr. Rajendran. And good afternoon, one and all present here. On behalf of the Department of Biotechnology, Faculty of Science and Humanities, SRM Institute of Science and Technology, Ramapuram campus, I am delighted to welcome you all at this wonderful occasion of the Skill Development Program on Molecular Inhibition of Endoplasmic Reticulum Stress by Vitexin in Post-Myocardial Infarction and in vitro and in vivo studies. I wholeheartedly welcome today's guest speaker, Dr. A. Ashok Kumar, Assistant Professor in Research at the Center for Laboratory Animal Technology and Research, Satyabama Institute of Science and Technology, Chennai. Sir, our sincere thanks to you, sir, for honoring our invitation.
Thank you,
warm
sir.
welcome to the, thank you. A warm welcome to the UG, PG students, research scholars, participants from various colleges and universities, my dear colleagues, friends, and students. The Department of Biotechnology at SRM IST Ramapuram campus is a 5-year-old entity in the Ramapuram. It having started in the year 2019, we began with an undergraduate program, and this year we have initiated the MSc program. Our faculty members have also received guideship for PhD programs, both part-time and full-time. To date, our faculties have published over 38 papers in various peer-reviewed journals with a cumulative impact of 200 plus. We have conducted more than 150 webinars, workshops, and skill development programs and have consistently organized national and international conferences. Additionally, we have signed for MOUs. As of now, 4 MOUs we signed with foreign universities, including UNISA in Malaysia, a clinical research laboratory, and an environmental industries. For the benefit of students, we have established 3 clubs within our department, namely Bioconsortium Club, the Journal Club, and a Biorhythmos, a non-technical club. These clubs provide platform for students to come together, discuss, and excel through their innovative ideas. With this brief overview of the biotechnology Department of Biotech, I conclude my speech. From the Department of Biotechnology, I once again welcome you all and wish this session great success in achieving its intended goals. Thank you.
Thank you, sir. Thank you for your energetic welcome address, sir. It's a pleasure and duty to introduce our today guest, Dr. R. Ashok Kumar. So that I call Dr. A. Nancy Jennifer, Assistant Professor, to introduce the guest. Madam, please.
Thank you, sir. Good afternoon to one and all. Myself, Dr. Nancy Jennifer. It's my pleasure to introduce today's chief guest to the Skill Development Program. The invited chief guest of the day is Dr. R. Ashok Kumar, Assistant Professor, Center for Laboratory, Animal Technology and Research, Satyabama Research Park, Satyabama Institute of Science and Technology. Sir has completed his postdoctoral research from University of Nebraska Medical Center, USA, between the year of 2020-2022. He completed his postdoctoral degree in biotechnology from University of Madras in the year 2019. He did his master's in biotechnology from Bharatiyar University and completed his undergraduate degree program in biochemistry from Manonmaniam Sundarnar University, Tirunelveli. His major area of research interest includes molecular cancer and vascular biology and molecular genetics. He has research experience and hands-on skill in cell line studies, CRISPR-Ca line method, mouse model, RNA sequencing and its analysis, and even more to report like. He has published more than 14 research journals, research articles in reputed and impactful journals like International Journal of Molecular Science, Applied Biochemical and Biotechnology, Clinical and Translational Medicine, Indian Journal of Pharmacology, Journal of Biochemical and Molecular Toxicology. He has also presented his research findings in many national and international conferences and symposiums. With this short intro, I welcome you, sir.
Thank you, ma'am.
Thank you, madam. Thank you for your nice introduction. Ashok sir, over to you.
Thank you, sir. Thank you for the introduction, ma'am, Dr. Nancy Jennifer. Before going to the topic, I would like to thank organizing committee, which means like organizing head Dr. Kamaraj sir, head of the Department of Biotechnology, and Dr. Dileepan sir, Professor and Head MCA, Vice Principal Admin, and conveners of this program, Dr. Rajendran and Dr. Nancy Jennifer, and Dr. Saravanan, Vice Principal Academics, for giving me this opportunity. And I have been worked for almost 5 years during my doctoral research. In this study, we were working with 2 different molecules like Vituxin and oligomeric proanthocyanidins. In these 2 compounds, we isolated from the Himalayan plant, that is Crataegus oxycantha, which is rich, which means like this plant have been widely used for a lot of heart-related disorders, especially myocardial infarction and atherosclerosis. These kind of conditions, people are using this Crataegus oxycantha. This kind of tablets, Crataegus tablets, which is widely available in European countries and US also. In India, it is available in the Ayurvedic formulations and in Siddha formulations also. So my topic of this session is molecular inhibition of endoplasmic reticulum stress by Vitexin, which is the major lead compound which is isolated from Crataegus oxycantha in post myocardial infarction, which is an in vivo and in vitro study. Sorry, small interruption in the system. Oh, okay. So coming to the non-communicable disease, the cardiovascular diseases and COPD, diabetes, and neoplasms like cancer, these all are coming under the category of non-communicable disease. There is two different kind of diseases are widespreadly available globally. There is a non-communicable and communicable disease. Out of these communicable diseases, CVD itself, it is affected almost 11.8% globally when compared to other diseases like diabetes and cancer.
Hello, Ashok sir.
1 minute, there is a small problem in the system. Just 1 minute, just a minute.
Okay, okay. Sir, Ashok sir, your PPT is visible, sir.
Am I audible? Rajendran sir, can you hear me?
Yeah. Your voice is audible. Your
Oh,
PPT
sorry for the
is visible, sir.
interruption. Okay, I'll continue from this. So, out of diabetes and COPD and cancer, COD has the highest rate of disease presence globally. So it is around 11.8%. And and all over the world, it is affected almost 41 million people. It is around 75% of global diseases. And moreover, out of this 74%, 77% of people are coming from the low and middle income countries. So it shows that the recent WHO data, it shows that CVD itself has around 17.9 million deaths all over the world. So it is the most dangerous disease when compared to even other diseases like cancer and diabetes mellitus. If you compare the overall global rate of presence of cardiovascular diseases, India, China, Russia, and USA has the high level of mortality rate in cardiovascular diseases. That's why a lot of researchers are going on to prevent this disease. How does these cardiovascular diseases are coming? There are different kind of risk factors which promotes the cardiovascular diseases. Some of our couch potato, which means sedentary lifestyle, like IT people are they are working in the same place, and it leads to the lot of health complication that leads to the cardiovascular diseases. And junk foods, liquor drinking, cigarette smoking, work pressure, and blood pressure, and sometimes gender also, which is affects one of the risk factor for cardiovascular diseases. Aging, obesity, and heredity also one of the major risk factor for cardiovascular diseases. What are the different types of cardiovascular? Which means that there are different types of cardiovascular diseases are there, like inflammatory heart diseases, cerebral heart diseases, rheumatic heart diseases, hypertensive heart diseases. Out of these diseases, ischemic heart diseases itself, which is affected around 46% of people globally. That is reported by WHO. In female also, it is around like 38% it is affected. So that's why ischemic heart diseases is the highest rate of mortality when compared to other heart diseases. What are the different types of heart diseases? Coronary artery diseases, atherosclerosis, myocardial infarction, left ventricular hypertrophy, and hypertrophic cardiomyopathy. In this topic, we are going to study about the how does myocardial infarction progresses and how we are going to reverse it. In normal heart, when if there is any myocardial infarction, means there is a sudden block of nutrition and energy to the cardiomyocytes, which leads to the ischemic injury in the cardiac cells. This ischemic injury further leads to the cardiac remodeling in the ECM. That leads to the irreversible changes in the cardiac tissue. How does this occurs? So coming to the phase 1, initially, if there is any oxidative stress in the cardiac cells, which leads to the apoptosis or necrosis in the cardiomyocytes. Coming to the acute inflammation phase, the injured cardiac tissue, which recruits the neutrophils, monocytes from the body, and directly it is converts its monocytes into macrophages. And further it leads to the secretion of MMPs and cytokines and chemokines, which leads to the fragmentation of matrix and inflammation in the heart tissue and vessel walls. And further coming to the phase 3, it further aggravates the angiogenesis process in the blood vessels and cardiac tissue, which leads to the transformation of fibroblast to myofibroblast and excessive production of ECM components in the tissue, which leads to the scar formation in the tissue. The scar formed cardiac tissues cannot be reversible to the normal one. So that is the dangerous place. So currently available treatments for cardiovascular disease, there are a lot of allopathy drugs which is available for cardiovascular disease, especially myocardial infarction. Some are categorized under nitrates and nitrates, that is amyl nitrite, nitroglycerin, isosorbide mononitrate. Beta blockers are proprapanol and metoprolol, atenolol. These are some kind of beta blockers. If you come to calcium channel blockers, nifimidine and nicardipine and dilcitabine and vermapiril. These are some drugs which is used for myocardial infarction. Coming to our plant, that is, we have chosen the hawthorn, that is Crataegus oxygantha. This is our botanical name is Crataegus oxygantha. It is coming under the family of Rosaceae. It is in India, which is highly present in Himalayas, especially Kashmir and Himachal Pradesh. In Hindi, it is termed as a Bansanli. And what are the major constituents which is present in the hawthorn are crataegoic acid, quercetin, kyperoside, rutin, vitaxin, monomeric catechogens, oligomeric proanthocyanidins, and proanthocyanidins. There are a lot of biological actives which includes antioxidant, anti-inflammatory, anti-arrhythmic, and anti-hypertensive, anti-lipidemic, and cardioproductive and hepatoproductive activities. The main pure compound which is isolated from the vitexin, which is an epicinin flavone, abundantly present in Vitex lucens kirk and Crataegus species like Crataegus oxycantha, mungbean and wheat leaves and chasteberry. Major biological activities of vitexin, which includes antioxidant, anti-inflammatory, anti-diabetic, anti-tumor and neuroprotective effects. This is the structure of vitexin. What are the major molecular signaling networks which is present, which is activated during myocardial infarction, which includes ERK mediated signaling and AKT-PA3K signaling, NF-kappa, TNF alpha mediated NF-kappa signaling, and Wnt beta-catenin signaling, and NRF2 signaling. These are the major signaling which is involved in the myocardial infarction. Apart from that, here we are going to study the endoplasmic reticulum and hyposignaling. What are the significant cross-link between the endoplasmic reticulum stress and hyposignaling? Coming to the endoplasmic reticulum is a major organelle for protein synthesis. It functions as a export of functional proteins to the other organelles and a translocation of proteins, and also it functions as a calcium hemostasis in the cells, and also ERAD, which means like it is unfunctional proteins which has to be eradicated from the endoplasmic reticulum. It functions through the action of proteosomes activity. Coming to the endoplasmic reticulum stress, UPR, which means unfolded protein response. Unfolded protein response, which means unfolded proteins which has to be immediately eradicated from the endoplasmic reticulum through the process of ERAD-mediated degradation of unfolded proteins. If after certain, due to high level of unfolded proteins accumulation in the endoplasmic reticulum, which leads to the endoplasmic reticulum stress. GRP78, which is one of the major molecule which is involved in the eradication of unfolded proteins by binding to the functional unfolded proteins, which means non-functional unfolded proteins. Once GRP78 dissociated from the unfolded proteins, this GRP78 will bind to the PERK, which is one of the surface receptors which is present in the ER, PERK, ATF6 and IR1. These are the three sensors for ER stress. Once PERK activated, it will activate the phosphorylation of EAF2 alpha to phosphorylated EAF2 alpha. This phosphorylated EAF2 alpha further will activate the ATF4. ATF4 is a transcription factor. It can activate the CHOP. CHOP is another one transcription factor for apoptotic genes. On the other hand, ATF6, it can further activate the cleaved ATF6. This cleaved ATF6, again, it can enter into the nucleus, thereby it can activate its target genes. On the other hand, IRE1 alpha, it is another one sensor for ER stress. This IRE1 alpha, once it will be like heterodimerized. Once it got phosphorylated, it will activate the normal X-box binding protein into the spliced one. This spliced X-box binding protein, it can translocate it into the nucleus. There it can activate its ER chaperones to the further aggravate the endoplasmic reticulum mediated stress in the cells. So we're coming to the hyposignaling, which is one of the major pathway which is present during the development of the cells. Here, this hyposignaling can be activated through the G protein coupled receptors or else some kind of EGF or FGF. A lot of surface receptors which can activate the endoplasmic, which can activate the hyposignaling pathway. During activation, this MST1, which is one of the major molecule which is in the hippo pathway, this MST1, it can directly enter into the nucleus or else it can activate the LATS1. This LATS1 further can activate the YAPTAZ. If once YAPTAZ activated, it has to enter into the nucleus. In nucleus, it will activate the cell proliferation. So when it is in the off condition, the YAPTAZ always will be in the nucleus, which means off condition means if hippo pathway is not in the activation mode. When YAPTAZ in the nucleus, it will activate its proliferation genes like BCL2, BCLXL. It will suppress the activation of apoptosis genes like BAGs and other caspases. So coming to the main theme of this study, how oxidative stress induces the endoplasmic reticulum stress in the cardiomyocytes. This endoplasmic reticulum stress further it activates the hyposignaling in the cardiac tissues. It further leads to the apoptosis of myocytes. So this is the main hypothesis of our study, like how vitexin inhibits mitochondrial dysfunction through the molecular crosstalk between the endoplasmic reticulum stress and hippo pathway during post-myocardial infarction. So there are two objectives we have taken. The objective one will be involvement of endoplasmic reticulum stress and hyposignaling in post-myocardial infarction. Objective two would be coordinated function of ER stress and hyposignaling in mitochondrial dynamics during myocardial infarction. Coming to the objective one, we have taken the in vivo model system and in vitro model system. And in vivo model system, we have segregated into 4 different groups. Group 1 served as a control, group 2 served as a drug control. We have given Vitex 1.5 mg per kg body weight. Vitex we have given 1.5 mg kg body weight for 4 weeks. And group 3, we have treated with the isoproterenol. Isoproterenol, which is one of the which is one of the antagonist of beta blocker, which can induce the myocardial infarction in the rat artificially. And group 4, we have induced the animals with isoproterenol, followed by we have treated the animals with Vitex for 4 weeks. At the end of the experiments, we have segregated the tissues and other blood things, blood samples, and we found that Hutchins staining clearly shows that isoproterenol induced group show the complete demolished architecture of cardiac tissues with high level of infiltration of neutrophils and monocytes into the tissue sections. And also we found that there is a edema and there is a complete collapse in the tissue structure. It was reversely in while we were treating with Vitex. And also we found that cardiac markers like CK and LDH and lipid peroxidation levels were significantly downregulated in Vitex treatment. And at the same time, we found that enzymatic and non-enzymatic antioxidants like vitamin C, vitamin E, SOD, catalase, GSH, and GPX also, which was significantly reduced during the treatment with Vitex. And coming to further to confirm the involvement of endoplasmic reticulum signaling, which means ER stress markers, we were conducted the PCR experiment. Here we found that mRNA expression of GRP78, ATF4, CHOP, Xbox binding protein 1, which was significantly elevated during ISO induction, which was further downregulated upon treatment with the Vitex. The same molecules were confirmed with Western blotting also. Western blotting also, which was showing the similar results like in PCR, which was also showed the upon treatment with Vitex, the expression of GRP78 and phospho-AF2 alpha was significantly downregulated. Coming to the
Hello, Ashok sir, what happened? Okay, sir, your PPT is visible.
Hello.
Yeah.
in between someone muted it, I think.
Hello, Ashok sir, your PPT is visible. Yes, sir, your PPT is visible, sir. You can start now. Ashok sir, are you there? Can you hear my voice?
Can you able to see the PPT?
Yeah, sir. Yeah,
Yeah.
it is visible.
Visible, okay. I will, okay. Hello, can you able to see the PPT
Yeah.
full
Yes,
screen?
sir. Yes, sir.
Okay.
Yes, sir.
Okay. So after conducting the in vivo study, we went to in vitro studies. In vitro studies, we have used H9C2 myoblast cells. Here, we induced the myocardial infarction in using the hypoxic chamber. In this hypoxic chamber, we were introduced nitrogen at a 95%, which means like 5% CO2, 95% nitrogen, and only 0% oxygen. So we were able to induce the hypoxic condition in the hypoxic conditions to the cells. We were kept cells for 6 hours for hypoxic condition. After hypoxia, we were reoxygenated cells with the normal CO2 incubator for 18 hours. In this process, we were grouped the cells into 4 different groups. Group 1 was served as a control H9C2 cells. Group 2, 6 hours hypoxia. Group 3, 6 hours hypoxia followed by 18 hours of reoxygenation. Group 4, hypoxia followed by Vituxin with treatment 60 micromolar and followed by 18 hours of reoxygenation. MTT assay was conducted to confirm the, which means like highest viability of Vituxin. We found that 60 micromolar itself showed more than 90% of cell viability, and followed by we found that when we are treating for, when we are kept for hypoxia and hypoxia followed reperfusion, the cells were showed highest level of DCF DCF-DA staining, which shows that increased level of oxidative, which means reactive oxygen species in the cells, which was significantly downregulated during treatment with Vituxin. Coming to the PCR analysis, we found that the expression of ATF4 and GRP78 CHOP and Xbox binding protein 1 was significantly downregulated during treatment with Vituxin. And the same results was replicated in the Western blotting also. We found that the expression of phospho-EA2 alpha and CHOP was significantly downregulated. And also immunofluorescence analysis also we were conducted. CHOP, which is once it get activated, it will be immediately it will be translocated into the nucleus. That's why we conducted the immunofluorescence. Immunofluorescence study also clearly shows that the CHOP was highly, which is translocated to the nucleus during hypoxia and hypoxia mediated hypoxia followed by reperfusion injury, which was downregulated during treatment with Vituxin. Coming to the Western blotting analysis, we found that the expression of MST1, phospho-MST1, YAP, phospho-YAP, which was significantly downregulated upon treatment with Vituxin. Sorry, upon treatment with Vituxin when compared to hypoxia induced, which means hypoxia and hypoxia reoxygenated treated groups. Coming to the apoptosis markers, we found that BCL2, BCLXL, which levels were significantly upregulated. At the same time, the expression of pro-apoptotic markers, cytochrome C and caspase-3, which was downregulated during treatment with Vituxin. So this in vitro and in vivo studies clearly shows that isoprotein induced animals and hypoxia reperfusion, hypoxia reoxygenation induced H9C2 cells showed increased ROS and decreased antioxidant status. At the same time, it showed increased expression of AF2 alpha, ATF4, CHOP, and also it showed the hyposignaling markers, thereby which induces the apoptosis. While treating with Vituxin, the expression of ER-stress markers and hyposignaling markers were downregulated, thereby which decreased the myocyte apoptosis. Coming to the objective 2, now we are going to check whether ER-stress and hyposignaling or coordinately induces the apoptosis in myocardial infarction. So in this objective, we have chosen the 2 different inhibitors, which is TUDCA, tauroursodeoxycholic acid, and solubilnar. Solubilnar is a selective inhibitor of PERK-mediated activation of ATF4 and CHOP. This signaling pathway, see, this pathway only will be inhibited by using the solubilnar. But when we are treating with the TUDCA, it is a non-selective inhibitor of ER stress. It can inhibit all the signaling, which means like PERK, ATF6 and IRE1. All the sensors will be inhibited. So coming to the experimental design, here we have taken the 600 micromolar of TUDCA and soluble as 30 micromolar. The same kind of experiments were conducted using the hypoxic chamber. Group 2 and group 3 served as hypoxia and hypoxia reoxygenation groups and followed by treatment with TUDCA and soluble. Here the same way we have conducted the animal experiment also. Here animal experiment, we have chosen the isopropanol as a induction chemical, and the group 3, we have induced the animal with the isopropanol, followed by we were treated with the TUDCA 400 mg per kg body weight at the concentration of 50 mg per ml intraperitoneally 24 hours before ISO induction. So we were treated the TUDCA as a pre-treatment, not a post-treatment here. So in this study, we found that activation of EAF-alpha, how does it is affected by TUDCA and solubenol, and how does it interact with hyposignaling. This both coordinated signaling mechanism, how does it affects the mitochondrial dynamics, and followed by apoptosis of myocytes. So here, before going to the experiment, we have fixed the effective dose of TUDCA and solubenol. Since a lot of, even though a lot of experimental papers which was available for TUDCA and solubenol, again we were conducted the MTT assay to fix the optimum dose. Here we found that 600 micromolar of TUDCA and 30 micromolar of solubenol, which was showed the effective dose for further experiments. And we found that when we were treated with the TUDCA and solubenol, which was effectively, which can be able to inhibit the ER stress markers to confirm the checking how does this inhibitors are working. So initially we were checking with the ER stress markers by using the Western blot and PCR analysis. We both the results showed that these inhibitors can able to inhibit the ER stress markers. After that, we further we confirmed this one with using the immunofluorescence also. This also we showed that soluble effectively it can induce the ATF4 mediated activation of CHOP. So coming to the hyposignaling molecules, when we are treating with the TUDCA and solubenol, when compared to TUDCA, solubenol, which can effectively inhibit, which can effectively inhibit the phospho-MST1 activation and phospho-YAP, and which can increase the YAP. Which means because this solubenol, which is a selective inhibitor for EAF2 alpha mediated ATF4 and CHOP signaling. So this EAF2 alpha mediated activation of ATF4 and CHOP, which can directly activate the phospho-MST1 and phospho-YAP, which is present in the hyposignaling. So further to confirm this one, TUDCA, which means like when we were treating with the animals, here also we found that it could be able to inhibit the CHOP signaling, but it can be able to, at the same time, it can be able to inhibit the phospho-YAP also. So we found that ER stress, which is directly activates the hyposignaling molecules, thereby which leads to the apoptosis of cardiomyocytes. So to confirm the whether this hyposignaling activation, which is directly induces apoptosis, or else it is this apoptosis is induced through mitochondria. So further to confirm this one, we have isolated the subcellular organs like organelles like mitochondria and cytosol. We will isolated from the cardiac cells, H9C2 cells, and cardiac cells, cardiac tissues. We found that the expression of phospho-MST1 and cytochrome C, which was highly expressed, which means like when you are treating with TUDCA, which was inhibited in mitochondrial group, at the same time, which was increased in the mitochondrial group, which was downregulated in the cytosol. Actually, whenever cytochrome C activated, which is once get activated, is coming to the cytosol, thereby which will activate the caspases, that which will lead
Hello, Ashok sir, are you there? Dear participants, please wait for a few more minutes. I think some problem in the guest laptop. So he will rectify very soon. Please wait for a few more minutes. I will share later. Focus later.
So am I audible, Dr. Rajendra?
Yes, sir. Yes, sir. Your voice is audible. We can see your PPT.
Yeah.
You can continue.
Okay. So coming to the conclusion, we found that the salubrinol and DUDCA, which can decreases the activity of hyposignaling through the inhibition of transcriptional activation of CHOP, and thereby which inhibition of hyposignaling, which inhibits the mitochondrial MST1 and mitochondrial membrane potential in the mitochondria, and which leads to the inhibition of myocyte apoptosis. So
Hello, sir.
leads to the which leads to the initial injury to the cardiac cells, which further elongated, which means further prolonged to the post-myocardial infarction. When we are treating with the Vituxin, which clearly which can inhibit the EA2 alpha ATF4 mediated CHOP activation, which further inhibits the activation of MST1 and PASPOYAP, and further leads to inhibition of myocyte death. And further, we found that this selective inhibition of EAF2 alpha mediated ATF4 CHOP signaling was further, which was confirmed by using the selective inhibitor like salubrinol and TUDCA. So this overall results shows that when we are treating the animals or cells with Vituxin, which inhibits the ER stress in the cells, which was activated during the myocardial infarction or else hypoxia mediated reoxygenation, and which also further inhibits the activation of hyposignaling, thereby which inhibits the mitochondrial dysfunction to promote the cardiomyocyte survival. Thank you all.
Thank you, sir. Thank you for your excellent presentation. Dear participants, if you have doubt or want clarification, you can ask now. And you can also post your questions in the chat box. Sir, we have one question. It is in the chat box. Sir, how do we selected optimal viability of TUDCA SAL since previous concentration shows higher viability?
Actually, we have chosen the minimal concentration, like we have not chosen the, which means our, when you are coming to the cell viability, you can choose more than 80%. Whatever the cells showing the more than 80% viability, we can choose any kind of dose. But when we are choosing here, which is the optimal dosage, which can inhibit the ER stress. So that's why we have chosen this dose.
Any question? Any question from participants? So, if no more questions, we move to vote of thanks. So, good afternoon all. I am Dr. G. Rajendran, Assistant Professor in Department of Biotechnology. On behalf of the Department of Biotechnology, SRM IST Ramapuram campus, I am glad to be standing here to express my vote of thanks. First of all, my most sincere thanks to the God for making today's event a successful one. I wholeheartedly thanks to our today guest, Dr. R. Ashok Kumar, Assistant Professor in Research, Center for Laboratory Animal Technology, Satyabama Institute of Science and Technology, Chennai, for accepting our invitation, sparing his valuable time, and sharing his science and research knowledge in cell culture and animal biotechnological field. Sir has explained elaborately the epidemiology of CVD, factors involved in the CVD, types of CVD, the role of Vitexin in controlling CVD, explained by in vitro and in vivo model systems. I hope all this information might be useful for all the participants to enhance your scientific knowledge and research. So, our sincere thanks to Dr. R. Ashok Kumar, hearty thanks to management, dean, VB admin, and VB academic for their constant support and encourage to organizing this skill development program. A special thanks to our respected HOD, Dr. M. Kamaraj sir, for being the catalyst that stimulated us to do the event like this. I would like to thank Dr. A. Nancy Jennifer. She is one of the convener of this program, and all my colleagues for their teamwork and effort in this event. A wide round of applause and thanks to all the participants who made this event a memorable one. Your participation, discussions, questions, and comments are always giving new ideas and encourage us to organize more events like this. So once again, my thanks to all the participants and expecting the same in all our upcoming programs. Finally, I would like to thank each and every one of you for being here and making this day a wonderful one. We will meet on another one program. Have a nice day. Bye-bye. So please switch on your video for taking the group photo. So feedback link we will post in the chat box. So it will be activated for next 10 minutes. So I request to all the participants kindly fill the feedback within the 10 minutes. So the photo real. So I request all the participants, please switch on your videos. Thank you. So we have posted the feedback link in the chat box. It will be activated for next 10 minutes. So I request to all the participants kindly fill the feedback within the 10 minutes.
Sorry, again, sir. Yes, sir. Card it with Google. Google. Again, and again, put the goal.
Dear participant, you will be receive your certificate within 10 to 15 days. So thank you for one and all joining this event. Thank you so much.
Please post the feedback link, sir.
Sir, feedback link is not there.
There is no feedback link. I guess. So please post it.
கொடுத்துருங்க நான் கொடுத்துட்டு ஒரு மாசம் நீங்க கால் பண்ணி தரேன் அதை பண்ணி தரேன்.
Sir, feedback link is not there, sir. Hello.
Is there any organizers?
Sir, வாங்க.
Hello, sir.
போலாம்.
Hello.
வாங்க. அனி 3:20.
Dear participants, the feedback link is also shared in the WhatsApp group. You may fetch from there and also fill the feedback. Kindly make a note of it. The feedback link is also shared in the WhatsApp group.
என்ன சொல்லுங்க? DRC ribonucleic acid. Come on. Started. Who started? ஆமா இது கவர்மெண்ட் அதெல்லாம் DRC DEOXY RC OXY